Welcome, and thank you for standing by. I'd like to inform all parties that your lines have been placed in a listen-only mode for the duration of today's conference. Today's conference is also being recorded. If you have any objections, you may disconnect at this time. I would now like to turn the meeting over to Ms. Diane St. Germain. Ma'am, you may begin.
Thank you. Hello, I'm Diane St. Germain, a nurse consultant in the Division of Cancer Prevention at the National Cancer Institute. On behalf of the NCI, Symptom Management Quality of Life Steering Committee, and the International Society for Quality of Life Research, I'd like to welcome you to today's webinar, Symptom Management Concept Design. Before we begin, I'd like to go over a few housekeeping details with you. To see the slides, you will need to have Microsoft Live Meeting installed on your computer. If you have not done this yet, please refer to the confirmation email or calendar invite for the links to download. Live Meeting will work best if you close all other applications. You will hear the audio part of the presentation by telephone at the number listed in the webinar invitation. You cannot hear it through your computer. If you need technical assistance, please dial *0. The webinar is being recorded. All lines are muted except for the speakers. The presentations and the slides will be posted and archived on several NCI and ISAQUAL websites. Information will be sent once they are posted, which will be approximately a month from today's event. You may use the question box on your screen to ask questions. You may type your questions at any time during the presentation, and we will read them aloud during the Q&A period at the end of today's presentation. You may open the Q&A box by clicking on the Q&A option at the top navigation on your screen and dragging it to the right-hand side of the screen. The objectives for today's webinar include to identify the required elements included in a symptom management concept, to identify key statistical considerations for a symptom management concept, and lastly, to describe issues to consider during concept development from a community physician's perspective. Each speaker will introduce him or herself prior to their presentation. I will now turn it over to our first speaker, Dr. Michael Fisch.
Thank you, Diane. Good afternoon. My name is Dr. Michael Fisch. I'm the chair of the Department of General Oncology at MD Anderson Cancer Center, and I'm co-chair of the NCI Symptom Management and Quality of Life Steering Committee. My job to get us started is to take you through an overview of this concept design. I'd like to start by providing some overall context by going over the organizational structure of the NCI Community Oncology Research Program called NCORE. So in the yellow box in the middle, you'll see that there are two basic components to NCORE. There are the community sites and research bases. The community sites are involved in facilitating enrollment to cancer prevention and control trials and working closely with research bases which are focused on the design and conduct of those trials, as well as the data management and biostatistics and dissemination pieces. Also involved are several divisions of the National Cancer Institute, including the Division of Cancer Prevention, the Division of Cancer Control and Population Sciences, the Division of Cancer Treatment and Diagnosis, and also one center, the Center to Reduce Cancer Health Disparities. Closely linked are the National Clinical Trials Network, where the treatment trials are conducted. And you can see the CIRB and CTSU are ultimately involved as well. This program is intended to include extramural investigators from NCI-designated cancer centers and other academic centers, including other kinds of research organizations. And the concepts that are put forth through the research bases are evaluated and prioritized through the steering committees. And in this case, there is a symptom management and quality of life steering committee that is in charge of this aspect. The steering committees are coordinated through what we call the CCCT, or the Coordinating Center for Clinical Trials. The responsibilities of the steering committee are to prioritize these trial concepts and also to do some other things, including convening state of the science meetings where critical questions are identified and prioritized, developing concepts for certain new trials utilizing task forces when appropriate, and periodically reviewing accrual and unforeseen implementation issues. The major goals of the steering committee are to evaluate and prioritize the trial. That's number one. But in addition, to provide expertise for the disease-specific steering committees. So, for example, there is a breast steering committee and a GI tumor steering committee and other steering committees where quality of life expertise is sought and is delivered through the Symptom Management and Quality of Life Steering Committee. Another major goal is to increase the availability of biologically plausible and feasible interventions to reduce cancer treatment toxicity and disease-related symptoms. The work of the committee is sort of organized through a constitution or standard operating procedures, and this is something that was developed by the committee in the spring of 2010, although the committee itself was first got started in 2007. The logistics of the committee are that there are a chair and co-chair. There is involvement of the different divisions of the NCI. There are representatives of the funded research bases. There are liaisons both for the disease-specific committees and also the investigational drug steering committee. And again, this whole steering committee process is logistically supported through the CCCT. It involves teleconferences for concept reviews. Periodically, there are face-to-face meetings as well, and from time to time, there are organized clinical trial planning meetings, and these are generally meetings organized around a specific topic, such as fatigue or chemotherapy-induced neuropathy or that sort of thing. When the NCTN reviews the work of the Symptom Management and Quality of Life Steering Committee and the other steering committees for that matter, there are certain criteria as they look through the whole body of trials that were approved. They're looking to see that approved and conducted trials are feasible, that they're clinically important, that they're making a scientific contribution. They're looking at the cost and resource commitment involved, and in particular, the unique suitability for the NCTN. So sometimes there is research that is considered appropriate and strong research, but it may or may not be considered uniquely suitable for this landscape. When investigators are putting together trials in symptom management and quality of life, there are really 8 broad, what I call key choices that investigators make. First, targeting symptom, deciding what they're going after. Secondly, deciding whether the goal is to treat that symptom or to prevent it, or some combination of the two. Third would be deciding about the population involved and whether the case mix of enrolled patients is to be something fairly broad or more narrow. So an example of broad would be patients with advanced cancer. Something more narrow might be post-treatment survivors after breast cancer treatment. Also a decision about whether a drug or non-drug intervention is to be used, and amongst the drug interventions, whether something like a natural product would be used, or a repurposed FDA-approved drug, or some other new targeted agent, specifically going after the symptom in mind or toxicity in mind. Choices need to be made about use of patient-reported outcome measures amongst the validated measures that might be available, and other validated tests or objective tests. The timing of the assessment is an important choice. There's a decision about whether or not to add correlative science elements to the study and whether they would be integral or integrated into the design. And finally, the overall study design, which might range from a feasibility study to a placebo-controlled phase 2 or even a phase 3 design. So a concept is basically a phase 2 or phase 3 study of at least 100 subjects. Again, it's developed through the research bases, but many times the lead investigators are coming from other academic centers, or ideas are coming from NCORE sites, what have you. So collaborations outside of these research bases are encouraged. The page limit is 10 pages, but it is recommended that these concepts be 5 to 7 pages. Things like consent forms and case report forms do not need to be included with the concepts. However, copies of all patient-reported outcome instruments do need to be reviewed by the steering committee in order to appropriately judge the concepts. The rationale involves stating what the condition to be studied is and that it's a common condition of great enough importance, focusing a background rationale on mechanisms that are likely to be relevant is very important, and comments about the feasibility and how likely it is for an intervention to be adopted by patients needs to be supported in the rationale, and really making the case of why this is an intervention uniquely suited to this particular landscape. The concepts involved a literature review that needs to be up to date, it needs to be highly relevant specifically to the proposed intervention in the proposed population. Results from previous studies need to be reported accurately, and the scope of all relevant data should be included, including some data which might not fully support the intervention. And logistical details like matching the reference to the text properly is important so that the steering committee understands and properly reviews the concept. The objectives and the hypotheses are critical. They need to be clearly stated, and all primary and secondary objectives in the hypotheses need to be outlined. These primary and secondary objective statements tend to foreshadow the patient-reported outcomes that are going to be used and the degree of change that is going to be considered meaningful. There needs to be fidelity to these objectives in other sections of the concept so that the whole document is internally consistent and clear to the reviewers. So now I will introduce Dr. Watkins-Bruner.
Good afternoon. My name is Deb Bruner. I'm professor at Emory and Associate Director for Outcomes Research in the Winship Cancer Institute, along with Dr. Fisch. I co-chaired the Symptom Management Health-Related Quality of Life Symptom Management Committee Steering Committee for the last 6 years and have recently stepped down from that position. I'm going to be talking about, I don't seem to have control, Diane.
Okay, I can advance your slides for you.
Okay. Next slide, please. Today I'll be talking about study design and some of the successful approaches we've seen in getting concepts approved through the steering committee. Next slide, please. So successful concepts clearly have common elements. And Dr. Fisch has already begun to describe some of these in terms of consistency. So the concept will have a clear and consistent focus beginning with the title through the background rationale and the objectives. The measurements or metrics must clearly map to the objectives. And as rational as that sounds, we find a lot of issues in that regard. The intervention has to be clearly explained, including the length of the intervention. And the statistical design must also, like the metrics, map to the objectives. And it must be appropriate for the metrics involved. The evidence to support the study needs pilot data to give us the prevalence of the event, symptom, or toxicity, which is the target. Now, there are two things that need to be stated when we're talking about the metrics or the outcomes. We generally see that investigators do a very good job on the first one you see listed here. We generally have investigators tell us by how much an intervention will be statistically significantly different from the control arm. However, on recent review by the National Clinical Trials Network, what we found is that our symptom management trials have largely been lacking the second step, and the bar is being set higher. So this is an extremely important topic and very timely. Not only is it important that investigators tell us the statistical difference on some metric. So for instance, that a patient-reported outcome will have a 5-point difference between Arm 1 and Arm 2, and that they propose that that will be statistically significantly different. We also have to be told or understand how that difference, that 5-point difference, will decrease the symptom, event, or the toxicity, and by how much. Then all of this is needed, obviously, for the sample size. And Dr. Naughton will speak more to that. Next slide, please. Other issues that have to be addressed are issues of treatment noncompliance or non-adherence, and previous problems with accrual in related or similar studies. If we recall that the definition of insanity is doing the same thing over and over and expecting different results, then we can propose to you that we frequently have an insane world that proposes the same accrual plans that ran into trouble in similar and related studies. Frequently, we see that investigators will tell us in the cooperative groups that, yes, there were accrual problems, they couldn't meet their accrual in a timely way or even had to close the study early, and that the plan is that they will have the same accrual process by sending out the study to their members and maybe emailing them a little more frequently. We propose that that is not enough, especially at the national level. The bar is higher here as well. We expect a significant plan to overcome noncompliance or treatment non-adherence and also accrual problems. The study needs to account for other treatments patients may be taking that will affect the endpoint of interest. Blinding is always important, especially when we have patient-reported outcomes and subjective endpoints. Now, for historic controls, we don't usually see this, obviously, in Phase 3 trials, but it is an issue, especially in randomized Phase 2s. This has been common in the past, but is becoming more difficult to justify with the frequency of changes in treatments. So time lag is a problem when you look at historical controls for comparison to the experimental arm. And there are usually differences in population these days for many reasons. So it's not to say that a historic control arm would never be approved, but again, there would be a significantly higher bar and significant rationale required to use that in a randomized Phase 2. Next slide, please. Now, successful concept designs across the phases, we don't do a lot of Phase 1 trials in symptom management, but we do some. I'm not going to spend a lot of time on this. The endpoints would be similar to treatment trials where we would be looking at toxicity and safety issues, usability, dose finding for the Phase 2. But if we do a Phase 1, it is incumbent upon the investigators to clearly walk us through how it would lead to a phase 2. I'm going to spend a little more time on phase 2s. Those investigators on the phone who have been at this a long time, we frequently feel that we know the landscape and have been doing this and have great expertise. The issue with that is based on the NCI portfolio review, we can say that the landscape is changing even for those of us who have been at this a long time and are experienced. Some of the ways it's changing is resource use for phase 2s. It is extremely important that every single endpoint and every single metric on a phase 2 and a phase 3 be justified. In days gone by, we used to collect a great deal of data, and sometimes phase 2s were treated almost like a phase 3 in the amount of data that was collected. Given limited resources, we can tell you that that cannot be the case moving forward, that phase 2s must be well justified in every single endpoint, every single metric, every timing of the endpoint. So gathering data over 15 time points, but only using the change score between baseline and 1 year does not fly anymore in review. You have to walk us through why all those other time points are necessary and statistically how they would be used. Not only that, in a phase 2, it has to be clearly defined as how each one of those data points will help in a go, no-go decision for the planned phase 3. So to reiterate, the days of collecting lots of data and hoping we will analyze them somewhere down the road, but we have a primary endpoint of, say, pre to post, is difficult to justify these days. Each endpoint, each metric, the statistical design for all must mesh, be consistent, and be justified. Next slide, please. Now, phase 3s. For phase 3s, again, we see a bar that's climbing. We have in the past done symptom management trials where we have not had good preclinical data on why an intervention would work. We have anecdotal evidence that it makes people feel better or seems to modify certain symptoms or toxicities. But previously, we frequently did not have preclinical data on the biological plausibility of why this intervention would work in this setting, in this symptom. That is getting harder to justify as well. We understand the challenges of obtaining preclinical data in the symptom management setting, and yet this is more of a concern at the national cooperative group level. So we may be using other portions of our network, including our cancer centers and the R01 mechanism, to get some of this biological data. But at the national level, biological plausibility is becoming a requirement in talking about study design. We also need to understand the choice of dose and the duration of the intervention. Again, pre-pilot data is required for phase 3 studies. Proposed intensity of an intervention is also required when we're talking about things like exercise or yoga. A side effect profile for our intervention is extremely important. Toxicity and safety never take a back seat. Interventions also may be more beneficial to a subset of patients. We frequently have all been there and understand the issues of trying to obtain ample sample sizes, but we need good rationale for broader samples, specifically if we see in the rationale that we think that the intervention benefits a subset of patients. So when we design a phase 3, it is extremely important, again, at this point, to point out the difference between national cooperative group trials and R01s. In R01s and our own program of research, our phase 3 trials may lead to future research. But at the national level, phase 3 trials are more and more being looked at not for how they lead to future research, but how they will improve patient outcomes, how national resources are being used in phase 3 to change clinical practice. Again, the bar is higher. Next slide, please. Issues of eligibility. When treating a condition, the population should actually have the condition and a level of the condition that would allow us to see a variance in response. I know that, again, sounds like common sense, but I have to tell you, at least in my own research, in my own research group, just by studying a condition, we have had the good fortune of seeming to cure it. Once we make a good rationale for a study, talk about the prevalence of the condition, we have gone to study things, and then seems that none of the patients have it anymore. By magic, we have cured it. We've had to change the eligibility criteria, lower the level of the condition to get patients to accrue, and then we have trouble with variance in response. We find that this has happened mostly because we have based the prevalence of the condition on literature reviews. It's becoming more incumbent upon national cooperative group trials to have preliminary data, not based on the literature or aggregates of multiple studies, but based on prior work within our own cooperatives with the patients to which we have access. And that gives us a more realistic rationale for the condition and the prevalence and the level of the condition in the patients to which we have access. Again, similar to what we're saying with the condition, if we are preventing a condition, then it is clear that the population should not have the condition if we're trying to do prevention studies. And again, we just talked about limiting the population to those who would likely have benefit. So we have an issue, again, with accrual, so we frequently see heterogeneous populations to boost accrual. However, if the benefit really is in a homogeneous population, a smaller population, we need to attend to that in the rationale. Next slide, please. In choice of endpoints, again, we want endpoints with high prevalence in the populations to which we have access, high severity, or that are highly important to patients' well-being. We have to clearly define the endpoints and how they will be measured, and we need to choose the best instruments for measuring endpoints. This isn't always as easy as it seems, and all of us on the phone are probably quite aware of that. We look at whether all the questions map to the objectives, to the condition for which the population is to have. And if the measurements are able to realistically be able to be standardized across sites, for instance, neurocognitive testing that might require credentialing. We also clearly look at the timing of the endpoints. Again, back to the point I made earlier, the days where we would have 15 endpoints, but a statistical design that did not include all of those endpoints are gone. We must have a statistical design that uses every measured endpoint and makes it relative to the decision-making in either clinical trials or future guidelines for patients or for moving on to the next study. We need to take into account the natural history of a condition and the mechanism of action of the intervention. And we clearly need to realistically account for potential loss to follow-up. Next slide, please. Further endpoint considerations include making sure that study on endpoints are timing is clear. Usually we like to see a baseline in relation to diagnosis or treatment, and then timing assessments at follow-up should be in relation to a fixed point. Patient-reported outcomes, although we're speaking to the choir probably on this phone, we all believe in them, may not always be the best or at least the only relevant outcome. For instance, in pain, it may be a dual outcome where you're looking at a patient-reported outcome of pain with a co-primary that looks at whether the pain medication on the intervention has increased, decreased, or remained stable. There are significant problems, and I think we have mostly gotten away from this issue, with the CTC as a primary endpoint. The NCI does not encourage this. It is a good measurement of adverse events, but not a good primary endpoint because it is not sensitive, has not been validated, and often double barrels questions, meaning there are multiple components to one question. We need to explain the use of all data. I've said this over and over. And obviously I'm saying this because it's one of the common issues we see in trials that come forward. And all measures must be included in the analysis plan. Next slide, please. The measurement tools themselves need to be explained well-defined. They need to be validated and validated in a similar population. So that provides issues that may need pre-pilot work because we still occasionally see metrics coming forward that were not validated in the population of interest. And we need to ensure that the primary endpoint maps well to the metric. So, for instance, if we're proposing to look at chemotherapy-induced peripheral neuropathy, and we propose that the primary endpoint is pain, but then the metric is a mixture of things like pain, numbness, tingling, quality of life, social well-being, we would find that a problem on review. The endpoints must clearly map to each objective. Next slide, please. And my final slide talks about this mapping. Years ago when I was in doctoral school, we actually had to go through actual visuals like this to map our conceptual framework to our objectives, to our metrics, and to our analysis plan. What we find is investigators who are not doing this either mentally or actually on paper find a disconnect between these issues. So to simplify this, I'm going back to how important this mapping is. I'm using the example of pain. Here we're just talking about mapping the objective to the metrics. If we have an appropriate metric for pain on the left, we may include things like frequency, severity, duration, intensity. We may look at it as a co-primary with or without or the stabilization of pain medication. But if we say that the primary objective is pain, but then include a metric that may be more appropriate for secondary endpoints that includes all the other things you see listed, that would give us pause at the review. And much of this can be discussed further in the statistical analysis section, and I will turn this over to Dr. Naughton for that. Thank you.
Hi, this is Michelle Naughton. I'm a professor at Wake Forest University. I am one of the co-vice chairs for the Health Outcomes Committee for the Alliance, and I hold a similar role in the Wake Forest Comprehensive Cancer Center research base in Winston-Salem. I'm joined here by my colleague, Dr. Dodd, who is in the Division of Cancer Prevention at the National Cancer Institute. Say hello, Dr. Dodd.
Hi, everybody.
Yeah. Dr. Dodd and I are going to take you through some of the statistical aspects of symptom management and quality of life concept proposals. I'll run you through the slide, and Dr. Dodd and I will both answer your questions at the end. At the concept stage, it's important to realize that the importance or purpose is to describe the statistical methods to be used to address the primary aims. And basically, in our view, the statistical section is where everything all comes together. All the things that Dr. Fisch and Dr. Bruner have talked about before, they're all come together. And hallmarks of a successful concept are really well-defined and justified specific aims. They're measurable, and very importantly, they have clinical relevance. You have a study design and measures that are appropriate to those aims. It's feasible to conduct the study in the community setting, and this is very important. Your power calculations are included, and they're accurate and aligned with the primary aims. And you have statistical analyses for your primary aims detailed in the concept. And you also have some discussion of some of the data challenges that you might encounter in your study and with the data you propose to collect. All successful concepts and proposals really are the result of a well-defined study team. You may have teams of clinical investigators, symptom management, quality of life experts, and so on. But statisticians in particular really should be included right at the very beginning of the concept development phase. Unfortunately, what happens typically is statisticians might be brought on midstream or toward the end of the development of the concept, and you really should avail yourself of the statistical expertise early on, right at the beginning of the concept development. This particular slide is meant to be kind of a checklist or a guideline for you to use in writing the statistical section. We're not going to go through every single bullet point on here because that will follow in the subsequent slides. But you can use this more as a checklist to go back and refer to when you're putting together your concepts for what to include in the statistical section. So let's go on and let's look at some kind of helpful hints for your specific aims. Some of the more successful concepts that we see are those that limit the study to 1 to 3 primary aims that are focused, justified, and importantly, measurable. You want to make sure that you power your study for all of your primary aims, not just 1, not just 2, but all of your primary aims. Similarly, you want to have well-focused and a limited number, perhaps 1 to 3, secondary aims. It's also important to indicate why your chosen secondary aims are secondary versus primary aims, and also why they're important to include at all. And from a statistical point of view, it's very important to make sure that unless these secondary aims are exploratory, that you have some study power left to test these aims after you're adjusting for multiple comparisons. In terms of your data analysis, you are going to need in the concept proposal an analysis plan for each of your primary aims. For your secondary aims, however, you only need to describe your general analysis plans for those aims that perhaps might be covered in maybe 1 to 2 paragraphs. The primary aims are a focus in the concept proposal. The statistical reviewers are going to look for certain things when they're reviewing the sections, and one of the primary focus would be to look to see if investigators have an understanding of the complexity of the study data that they're going to be collecting. They're going to be looking for such words or buzz phrases such as how you're going to handle missing data, multiplicity, blinding, data transformations, and so on. And just a couple words about data collection. Most likely, these types of how you're going to collect the data, the metrics that are going to be used will be covered in the methods section. But what we need to know for the statistical section is the feasibility of collecting the data using the methodology you propose. And why you need that is that we have to have estimates of how many completed surveys, samples, whatever it is at each time point you are expecting to complete. So we need to know what the attrition might be across the data points. This would influence your analysis plan, but it also has relevance for your sample size in terms of what your expected attrition might be during the course of the study. Dr. Dodd and his colleagues at DCP and CTAP put together a list of some of the common problems they see in concepts they review. Some of the most common are power calculations that are included that are missing or not included, or they might be included and they're inaccurate. You might have a power calculations that included for only one versus all of your primary aims, and that's very important to make sure that you have sufficient power for all of your primary aims. Another problem might be you list multiple secondary aims, but you really don't talk about if you have study power that's sufficient to study those aims, particularly after adjustment for multiple comparisons. You might also see, or kind of like a laundry list of secondary aims that are included in a concept, but they have very limited justification. And as Dr. Bruner just mentioned, you really now need to make sure that you are not proposing to collect any kind of data or have any type of aim unless you are, you can tie it back to clinical relevance, good study justification, and have analysis plans of what you are going to be doing and how you're going to be analyzing that data. Other problems could be things related to data analytic techniques that are not appropriate or they're not really optimal for the specific aims and data to be collected. You may also have no discussion at all of any of the potential data challenges and how they will be handled. And in any study, you're going to have potential data challenges, and so it is important to indicate how those will be handled during analyses. And one of the pet peeves among the statisticians is when investigators take a continuous measure and they turn it into a categorical variable, which really just truncates the range and eliminates data or cases. An example of this might be if you have a single item measure that asks the patient to say, how would you rate your overall quality of life on a scale from 0 to 10? And you just decide for analysis purposes that you're just going to look at people who score like a 4 and under or 5 and under and so on, instead of using the entire continuous measure. So just something to keep in mind. The next kind of common problem is looking at what's a clinically meaningful difference. And this is really a nemesis for a lot of us who do symptom management and other types of clinical trials. Oftentimes what is seen in a particular trial is that investigators will put in that they expect to see a half of standard deviation unit change, and that will be clinically significant. And the issue is why. It really isn't sufficient to just put in the half a standard deviation in the analysis plan with no justification. For example, did a prior study use this unit change based on some empirical work? Did you have some pilot data from which you got this half a standard deviation? Now, there are certainly many patient-reported outcome measures that have been calibrated to a half a standard deviation. If that is the case, then you need to cite references and other support for that. It could be the case, too, that you might have a secondary aim where you're actually looking to explore what the clinical significance might be, and you might be collecting data to calibrate a measure with some clinical outcome in the study you propose. Indicate that in your study and also how you plan to analyze those data. But more often than not, many of us don't actually know what a clinically meaningful difference might be for certain types of outcomes. And if you don't know, be honest and provide a rationale for the units of change that you have chosen. But do not leave it out. You need some type of a justification for this. And just a note in general, err on the side of explaining more about the choice of the primary and secondary aims, the study overall rationale, and the planned analyses. You need to think about this in terms of who your audience might be. You only have 10 pages for the concepts. But we believe that these types of points are very important and that you should spend some time on these. That's been also mentioned by Dr. Fisch and Dr. Bruner earlier. You need to make your case for your specific aims. You need to start with what is known in the intervention area, what your study would do to further science and impact clinical care, and then funnel this down to perhaps 1 to 3 very specific and targeted primary aims. In general, know your audience. Statistical reviewers, like many of us who will review studies, we often do not have a thorough understanding of the patient population or the intervention or the disease or the treatment that's being proposed. And just like if you were writing an R01 or an R21 or any other type of grant, it's your job to be clear in your study background and justification so that everyone who is reviewing the study can follow your rationale and your study design. So in general, all reviewers really want to be convinced that you have relevant and testable study aims, your sample size and power estimates are correct, it's feasible to recruit and retain your participants, and very importantly, that you can collect data in a community setting. You have well thought out patient safety and confidentiality. Your data analytic techniques are optimal and appropriate. And you have an understanding of the strengths and weaknesses of the study design. And you've outlined how the major weaknesses will be addressed. So our major take-home message to you is, number 1, find a statistician to assist you. Do it early. Do it often. And number 2, go back to number 1 if you haven't found a statistician yet. I'm now going to turn you over to Dr. Judith Hopkins, who is going to give us some insight and perspective from the community sites.
Good afternoon. My name is Judith Hopkins, and Jeff Kirschner and I are the community oncologists on the NCI Symptom Management Quality of Life Steering Committee. I am in practice in Kernersville, North Carolina with Novant Health Oncology Specialists and am the co-principal investigator of the Southeast Cancer Control Consortium. Next slide, please. It is critically important to have an experienced community oncology researcher on your study team from the beginning. They can help you determine if the equipment or procedures necessary for your clinical trial are available in the community setting. They can help you avoid financial hardship for the patient and the practice. Innovative studies will often require tailor-made recruitment strategies, instructional webinars, or face-to-face training of study coordinators, and need to be planned and budgeted for. Next slide, please. Remember, most community oncology practices are only open between 8 to 5 Monday through Friday. Studies that require minimal office staff and oncologist involvement, with study visits aligned with standard treatment guidelines and have short follow-up, accrue the best and are the easiest to complete. Next slide, please. Timely protocol completion occurs when the trial answers important clinical questions or solves vexing quality of life issues and grabs the interest of both the oncologist and the patient. Jeff will next describe examples of successful and challenging protocols in the community setting.
Thank you, Judy. I'm the Director of Research in Syracuse, New York at the Hematology Oncology Associates of Central New York. I'm the Principal Investigator for our CCOP, and as Judy mentioned, I'm one of the 2 community representatives to this committee. I'm going to mention 2 of many protocols that have both been successful and unsuccessful. The first one, which was very successful, was URCC 07079. The disclosure is that I was the study chair and Judy was a co-investigator. It was designed to see if we could prevent pegfilgrastim-induced bone pain. It was a randomized control study of naproxen. It's a very frequent problem that patients experience. It's clinically important. It's of great interest to oncologists, nurses, patients, and families. It's easy to identify and randomize patients with really minimal physician involvement. The nurses and the research associates do most of the work. Next slide, please. There was straightforward randomization. The treatment was relatively easy to do. There was relatively short follow-up, and we accrued over 500 patients in 1 year. And one of the reasons this protocol was successful, as prior speakers mentioned, we had both community oncologists and statisticians involved from the get-go with help from academicians, people that had PhDs and knew the scientific process even better than we community oncologists. Unfortunately, there have been protocols that have had to close early and never came to completion because it turned out they were really not practical to do. One such example is on the next slide. This was RTOG 0123. And radiation-induced pulmonary toxicities are really an important clinical problem. And when this study was designed, it was felt that it was doable. It was a phase 2 randomized trial with relatively low numbers of patients required, studying captopril in patients receiving radiation with or without chemo for limited small cell or stage 1 non-small cell lung cancer. As you could see, there was a lack of prospective feasibility assessment. Eligibility was more difficult than was thought ahead of time. The patients and physicians weren't as interested as the investigator thought. But the main reason for its failure was there was really a lot of work required by both the investigators and staff. And I'm not trying to suggest that we not do protocols because of the excessive work required, because there's some very important scientific questions that need to be answered. But perhaps investigators need to be innovative and get outside funding or other help, because community oncologists have limited time to devote to this type of research, and anything that's really complicated is going to be difficult to carry on. Next slide, please. So that's basically what I was going to say about 2 sample protocols, and I'm going to turn it back over to, I believe, Diane.
Well, thank you very much. This is Mike Fisch. I'll go ahead and take it from here to begin the question and answer session. Of course, everybody's audio is muted, so people are not calling in. But the way you can submit questions is by going to that Q&A box on the right-hand side of your screen, or maybe it's, in my case, it's on the left-hand side of the screen. But find that Q&A box, expand it, and type in your question, and then we can see the questions and go from there. So I'm going to start with a question and address this to Dr. Bruner, and that'll give Dr. Bruner a chance to come off mute there.
I'm here.
The question is this. It's very frequent that we want to measure an outcome for which an ideal instrument doesn't really exist. For example, a PRO which has been perhaps well validated and has an established minimum important indifference, but from which there are limited data in the precise population that you want to study. And the comment also describes the fact that sometimes the timing of the studies or the exact nature of the patient population is opportunistic and driven by the disease committees, and the quality of life investigators are trying to find that PRO. Any advice about that?
Well, we've done a couple of different things. One is that some of the cooperatives have used the PROs as secondary endpoints and then have actually done validation study within that trial, which works for larger phase 3 trials when you have enough of a sample size to do validation. We've also looked at this very rationally. We do have a trial open currently in women who are getting pelvic radiation therapy, and we're using an instrument, a patient-reported instrument, validated in men for bowel and bladder issues. When the Symptom Management Committee reviewed this, it was not validated in women, but we looked carefully at the fact that the symptoms mapped really well to the objectives, and we had no plausible reason to think that the questions would have been different in men or women for some of the bladder and bowel issues that they were looking at. So it's one of two ways. There can be concurrent validation if the sample size is large enough, or if we can have really rational discussions about whether the endpoints really map well, then it may be okay.
Thank you, Dr. Bruner. So another question has come up, and this one's for Dr. Kirschner. Dr. Kirschner, can you give an example of what caused the excess work for oncologists in that second example that you described in the webinar?
Well, I wasn't participating in that study per se, so I can't really address that. But examples in other studies have been really complicated consent forms, screening patients that may or may not be eligible excessively and then finding out that they weren't eligible. Some protocols require 5 to 10 forms to be filled out at each visit, and sometimes the visits are excessive. And as a member of the committee, we've pointed that out to investigators. Sometimes blood draws are excessive. So there's a lot of factors that go into making a protocol unsuccessful. And in addition, long follow-up. Long follow-up is a deterrent often. But again, it's just one factor. And I don't think any of us turned down a really clinically important study based on any one factor. But when the factors combine to make it unfeasible, sometimes we don't even activate the protocol.
Thank you.
So
the next question I have is does somebody else want to comment on that?
Yeah, this is Melissa. I just wanted to, and I'm helping to moderate this event. I just wanted to repeat the instructions for asking questions. The Q&A box, you have to open it first before you can drag it to one side or the other. It's on the top navigation bar, and it says Q&A. So if you click on that, you can type your question in, and when you type your question in, click on the word ask rather than the hand, because that will publish your question for us. Thank you.
Thank you very much. That's helpful. So the next question I have is for Dr. Naughton. So you mentioned in your part about the statistics the idea of calibrating an outcome to a 1/2 standard deviation. Can you explain a bit more about what you mean here and provide an example of what calibration means in this context?
I may have Kevin answer this question, if you will.
Sure, Kevin, you want to join in and explain this notion of calibration to the 1/2 standard deviation change, what that refers to?
Well, mainly the idea is that even though it's possible to do a power calculation using just nothing but what we call an effect size, and that's just like a fraction of the standard deviation of the outcome that you want to do, that I think the important thing there is that even though that's all you need to do, and although a lot of the literature that Cohen puts out there says that half a standard deviation is usually clinically meaningful, I think the important thing is that we'd like to see as a statistical, we'd like to see what that really turns out to be. So if possible, we'd like to see some sort of literature review or some sort of other studies that says this half an effect size is, or half a standard deviation is this many units. Don't just say it's half an effect, it's half of a standard deviation or effect size of a half, but actually say from other studies or something else, we think that it means this much. And so have a mapping between some actual differences in the actual metric that you're using, rather than just sort of an overall effect size is really, I think, the important thing.
So are you saying that it might be something like a 5-point change in this scale is roughly equivalent to a 1-level change in performance status?
Yeah, for example, something like that, where you actually kind of hang it on real life change in the metric, as opposed to just saying we think that we don't know what the change is going to be. We don't know what the actual change is in the actual metric, but we know that if there is a change that's big enough, we find it in our power calculations, and that leaves us wanting a little more.
Okay.
See the distinction there?
Sure. Kevin, while I have you, let me ask you another question. Dr. Naughton's slides referred to a couple of terms that are commonly seen in protocols and statistical reviews. One is the word multicollinearity, and the other is clustering. And I wonder if you could just give a non-statistician's brief description of what these things really mean and how investigators should sort of recognize when they need to think about these things.
Okay. Was it multicollinearity that was the thing that you flagged, or multiplicity?
Multicollinearity and clustering.
Okay.
Two different words. Statistical jargon.
Well, multicollinearity just means that if you have several different outcomes or things that you want to control for, if you're going to say, I'm going to look at quality of life, but I want to adjust for all these different things, if your adjustment, if the things you want to adjust for, if they are all related to one another, it's really hard to tease apart what's important. And so if you actually have, especially if you have, for example, two quality of life measures that are highly correlated, and you put them in the same model together, it's going to be hard to figure out which one is driving the change in the metric, the outcome. So that's basically it's something that is common, that if you're going to put a lot of if you're going to try to adjust for or test the independent effects of lots of variables, if they're related to one another, that causes some statistical problems. And so that needs to be more fleshed out. And the clustering of responses generally refers to things, if you're not going to have random groups of individuals or random individuals, but instead you're going to randomize at, say, the clinic level, where say everyone at a given clinic might be getting the same sort of treatment, then you have to take that into account in your power calculations, because the thing that drives your power is not how many people you get, but in this case, how many clinics you get, because if the people in the clinic are all getting the exact same treatment, you're really looking at a clinic level observational unit instead of a person level. And there are ways to do that. There are ways to do power analysis for what they call cluster randomized trials and things like that. So there's stuff out there to do that. There is one more thing I do want to mention that is a key word that we've been we've been wishing we'd see more of, and that's the discussion of multiplicity. That's when you have multiple outcomes, how to, there needs to be, it would be great if there were some attention paid to the idea that if you've got 3 primary outcomes or 2 primary outcomes, you don't power your study to test one of them at an alpha or a p-level or an alpha of 0.05. At the very least, you need to do something like say, okay, I want to spend some of my power of my study or spend some of my alpha level on this outcome and some on this other outcome. So doing things like a Bonferroni adjustment, for example, with 1 or 2 things, goes a long way toward sort of putting our fears at ease that you're actually trying to power the study to answer all of your aims at once, which came up several times in the different presentations. So that's another important word to kind of ask your statistician about, that the one that you've gone and gotten, right? You've all gotten one, right? So,
okay. Well, thank you. I think that's an interesting way of thinking about it, almost like you have a budget of money and you're having to sort of discount or sort of spend your money a little bit differently if you actually intend to equally weigh 2 primary outcomes.
Yes, exactly.
Way of explaining it. So here's another question. This one is for Judith. So Judith, can you explain how you participate in steering committee concept reviews? What role do you often play, or how do you come into play in the review process as a community oncologist?
Well, I have the option of reviewing the protocols primarily, which comes about as a rotation with other members of the committee. But then I also review the protocols with an eye toward whether or not they would work in my center. And as Jeff mentioned in his talk earlier, I look at whether or not it can be easily done in the clinic setting. Is it going to interrupt office flow? Is it going to require a lot of time from my staff, or can it all be done by my clinical coordinator? I look at whether or not the patient's going to have to come back for multiple visits that are not coordinated with their standard of care. But the key thing that we look at is whether or not it is going to improve the quality of our patients' symptom management.
Great. Thank you. So another sort of a logistical question, and I'll address this to Dr. Bruner. Does the study team or lead investigators actually talk to the review committee at any point in the process?
Yes, there's a few minutes, 5 to 10 minutes prior to the review where the study team may come on. What happens is that the symptom management review committee reviews, obviously, the concept in advance, and at least 1 day prior sends questions to the investigators that have arisen from the review. And it's just a few questions. It's the high priority questions, or the questions that come up most commonly. They are sent to the review, to the investigator and the investigative team. So they have at least 24 hours to prepare a response. They're asked on the phone to the symptom management committee. The symptom management committee focuses on those high priority questions, gives them a few minutes to answer, and then they are recused as the rest of the review continues.
Great. That's very helpful. So I have another question that I think I will address to the whole group, maybe Dr. Bruner to start with, but maybe others will have a point of view. And this has to do with the importance emphasized during the webinar on discussing physiologic mechanisms. The question says, some of the symptom management interventions may work by changing, quote, cognitive mechanisms, how individuals, say, think about their symptoms. Is this an acceptable or appropriate content to include in the concept write-up? And do any of the panel have any advice about that?
I'm not clear on the question. If they're saying that if the intervention is proposed not to change a symptom, but to change a perception of a symptom, I cannot say that on review of the national portfolio that would have been a high priority study for the cooperative groups. It may indeed be an important and rigorous question, something that would be good for an R01, but the national portfolio is looking at actual changes in mechanisms, decrease of prevalence, or minimizing severity of symptoms.
Yeah, I think the whole question is it's tricky, but I agree with your response, Dr. Bruner. In fact, it sort of made me think about this instruction that maybe some of us got in medical school when we're prescribing, let's say, a tricyclic agent for depression back in the old days, and somebody might explain that to the patient that when your mouth starts to get dry, then you'll know that the drug is getting into your system and beginning to help you. You know, the way of framing a side effect. So I can imagine, you know, some sorts of interventions that reframe or otherwise help patients think differently about their lives or their symptoms in a way that could be impactful. And, you know, is that a mechanism, and how would you demonstrate the mechanisms? What you usually see with concepts like that are conceptual models and conceptual underpinnings for the approach. But you know, how that might be reviewed and prioritized in the system, I think is you know, is hard to say. I think a lot of the mechanisms of symptoms and toxicities tends to be thought about as molecular or biological mechanisms, but that may or may not be the only thing that gets prioritized. It depends. Anybody else want to take a stab at that?
Yeah, I was just a little bit confused about it. Is it to look at kind of, for example, the neurocognitive pathways, changes in pathways, or is it to look at such things as like cognitive restructuring, which might be more of a behavioral outcome? My first thought on listening to the question was, were they talking about behavioral change models? But it sounds like that they're also looking for physiological changes as well that might occur for those. And I do recall seeing some protocols where we'd at least be looking at maybe some predictors of behavior change or other types of physiological measures that might be related to that. But I don't recall any types of studies that I think are addressing what the individual was asking.
Okay. Let me get to another question, and this is about statistical analyses, and maybe Dr. Naughton, you can address this also.
Okay.
Are statistical analysis plans required for exploratory objectives or endpoints?
Well, it was one thing that was specifically covered in the slide set, that you need to have some general analysis plan that would be put into this. An exploratory objective or an endpoint would be a secondary outcome in the studies, and so you need much more detail for your primary aims, but not for your secondary or exploratory endpoints. That you can't ignore it. You need to give some indication of maybe what statistical techniques and methods that you might be using to explore the aims or the objective. I don't know if Dr. Dodd has some other comments.
Yeah, I'll just add another couple of things. I think if you go along with one of the earlier slides, err on the side of giving more rather than less in your concept. And it's great to have statistical analysis plans for secondary endpoints and for exploratory aims. I think usually what we're looking for is say in a phase 2 study where you are trying to think about how you're going to move forward for phase 3. If the exploratory endpoints are trying to get it, something that you're going to use, you should give some thought to how you're going to analyze it. Give things like a confidence interval around what you think you can find, so that you can say, okay, if I do this with this many patients, I'll have, it may not necessarily need a formal power analysis, but you can say, I think I'll be able to get my answer to this exploratory aim or this exploratory measure. I can get that to within this many units, with a bound of that. So just to kind of give a little sense of is it going to really help in going forward to the next phase of the trial. I mean, the phase 3 trials, all those things are supposed to kind of end up and say, at the end of my phase 3 trial, I'm going to know everything there is to know about this particular setup. So I think the having exploratory, lots of exploratory aims in a phase 3 is not really what a phase 3 is for. But exploratory in a phase 2, I think they're more acceptable.
Okay, I think that's helpful. So some sort of statistical statements provides a sense of the quality of the exploratory look, it sounds like.
Yeah.
So here's a sort of a follow-up question that I'll maybe try to address. The follow-up question was, the perception of a symptom could be considered self-report, such as pain or cognitive changes. Would this not be of value? And here, I think that this question might be getting at our discussion about mechanisms, and I don't want to confuse people to think that self-report, patient-reported outcomes are not considered valuable. But here might be an example of a mechanisms issue. So and I'm just going to make something up totally out of the blue, but let's say that people observe that patients who are getting metformin for their diabetes or hyperglycemia and who also had ringing in their ears had resolution of that symptom. The tinnitus disappeared in the people who are taking metformin. And based on that observational data, somebody wants to propose to actually prescribe metformin to people who are bothered by ringing in the ears, tinnitus. So the self-report of ringing in the ears would be valid. But the committee would be highly interested to know what the proposed mechanisms might be, so that, if the trial were negative, it's not a completely valueless trial. And if there's no proposed rationale for why metformin would reduce ringing in the ears, then that trial may have trouble going forward. I think that might be at least a hint about what proposed mechanisms questions are really getting at. Dr. Bruner, what do you think? Does that explanation make sense from your experience on the committee?
I agree, Mike. I don't really have anything to add to that. I think that example is good.
Okay. So I have a question for Jeff. Jeff, how do you interact with research bases when it comes to designing and thinking through these trials? What role do you have as a community oncologist at that level?
That's a good question. And I could describe my own experiences with the Alliance. Can you hear me? Yeah. Anyway, I can describe my own experiences with the Alliance and several research bases such as URCC. Community oncologists are involved from the onset. We have members on most of the disease committees such as symptom intervention and prevention and other cancer control committees. There's a member from the community and from the CCOPs on the executive committees and on the concept review committees within the cooperative groups. At least in the Alliance, we have a community oncology member as a protocol co-chair of every study that's done. So there's input from the onset in terms of what needs are out there and whether the study can be done promptly and whether there are barriers that can be overcome. In addition, some of the community members actually write the protocols or get involved in concepts based on their clinical experience. Judy and I are examples of that. We've both recognized some problems that our patients were having and developed protocols with the help of the research bases and carried them forward.
Thank you very much. I think that's such an important point about the role of community oncologists, nurses, and other kinds of providers who are involved in cancer care at different levels. They all have the opportunity to not only interact with the research bases, but provide leadership on protocols and authorship, and sometimes first authorship on publications, et cetera. So it really is an integrated system of participation. So I think it's about time to wrap up, and we've run out of questions. One of the questions that commonly came through was a question about how to access the slides, and so I'll turn it back over to Diane, and Diane, maybe you can address where people can review the webinar, recommend it to others, or get access to the slide materials, as well as other housekeeping issues.
Several NCI websites, including the Division of Cancer Prevention, and they'll also be posted on the ISAQOL or the International Society of Quality of Life Research website. It'll take about a month before they're posted, and the slides will be posted along with the audio version of today's webinar. So if you have colleagues that didn't have the opportunity to listen in, you can pass that information on to them. And if you'd like to have this as reference for yourself in the future, they'll be available.
Diane, can you back up just for a moment? Because I think I heard you sort of midstream as if you came off mute or your sound picked up a bit late. What was the very first portion of what you described there about the slides?
Thanks, Mike. So the slides, along with an audio recording of today's webinar, will be available on a variety of NCI websites, including NCI's Division of Cancer Prevention. And they will also be on the ISAQOL's website, the International Society for Quality of Life Research. It'll take about a month before those are posted, and we will send out a reminder email to you all to let you know when they are indeed posted, and you can share those with other colleagues and refer to them at your convenience. The other thing you'll be receiving is a short survey regarding today's webinar. If you could please take a few moments to complete that, that'll give us an opportunity to plan future webinars, get a sense of what you found helpful, and ideas for content for future webinars. So in closing, I'd like to very much thank the speakers today for their wonderful presentations. I'd also like to thank several people, Sue Rossi from CCCT, Yvette Ortiz, also Melissa Glim and Catherine Jenkins. They're from the Office of Communication and Education, and they have worked very hard and diligently behind the scenes to make all of this happen today. And lastly, Dr. Warder McCaskill-Stevens for her support from the Division of Cancer Prevention. So I will close and thank you, the participants, very much for joining us today and for your participation.